Hello, I'm Dr. Marwan Sabah. I am professor of neurology in the Alzheimer's and Memory Disorders Division of the Barrow Neurological Institute in Phoenix, Arizona. Today, this session is titled Back to the Future, Emerging Opportunities to Treat Basal Forebrain Cholinergic Neuron Dysfunction in Alzheimer's Disease and Lewy Body Dementia. The questions that we need to consider in this symposium is, the first is the obvious one. Why should we be revisiting the basal forebrain cholinergic neuron system? We know that BFCNs serve a modulatory role in cognition by optimizing cortical information, and studies suggest that it has many projections into multiple areas, including limbic structures and cortical structures, that drive a variety of cognitive abilities. Data going back many decades now suggests that the loss of colon acetyltransferase staining in neurons has been seen. In fact, if you look at the earliest data, the CAT staining was literally 1/4 or even 1/10 as prominent in Alzheimer's disease as it was in young aging, suggesting that the cholinergic deficit was an early and seminal event that led to cognitive decline. The question then and larger is, does the BFCN system play a convergence? And that is to say, when we talk about the convergence of neuropathology, meaning amyloid, and cognition, meaning mediated through acetylcholine, the neurotransmission, how do these two events converge? And the answer is through the BFCN system. The cholinergic hypothesis is basically talking about the loss of acetylcholine, but that is not just a general principle of neurotransmission. That is driven and mediated by loss, particularly in the BFCN region, and that the centers of cause progressive loss of limbic and neocortical cholinergic neuroinnervation. Neurofibrillary degeneration in the basal forebrain is believed to be the primary cause for dysfunction and death of forebrain cholinergic neurons, giving rise to a widespread presynaptic cholinergic denervation. The reason this is so important is that the earliest treatments were based on this understanding. And when you look at cholinergic cholinesterase inhibitors, going back to even before we had them, people like Giacobini were talking about this idea that improving cholinergic function would be very useful, and has been and continues to be one of the mainstay treatments in Alzheimer's disease therapeutics in dementia, and they're used often in Lewy body dementia. The cholinergic deficit is present and is very well identified in both Alzheimer's dementia and Lewy body dementia, but is actually more severe in Lewy body dementia. The other thing that many of the audience may not understand is, beyond the cholinesterase inhibitors, there have been attempts to target the BFCN system, and a company called Serogene was looking at an adenovirus vector to inject into the brain to deliver an NGF stimulating treatment and trophic factors but it never advanced beyond early investigations. These data lay the groundwork for our symposium today. And the first symposium is from Professor Ole Isaacson from Harvard Medical School, and he will talk about the contribution of BFCN dysfunction and degeneration to Alzheimer's disease, and particularly also in Down syndrome, showing that amyloid is an important convergence. We know it's a neurotoxic properties, and it has its particular effects, as Dr. Isaacson will show, in the BFCN system, and that is probably the driver of the clinical phenotype. So the amyloid biology drives the clinical phenotype through the dysfunction of the BFCN system. Professor Nixon will be talking about RAB5 and the signaling endosome NGF factor, and how that affects the mechanism of BFCN dysfunction and degeneration in preclinical results with novel treatments approaches based on the signaling endosome RAB5, which can affect the NGF receptor. Our third presentation will be by Dr. John Allum, CEO of EIP Pharmaceuticals. He's going to talk about clinical results with novel approaches that reverse BFCN dysfunction. There are now a few small molecules that are in development that have been able to show robust clinical signals in Lewy body dementia, which is very exciting because we've not had a lot of successes in Lewy body dementia, and he's going to talk about two that directly affect bfcn dysfunction. And with that, I introduce Dr. Isaacson.
My presentation today will deal with the Back to the Future on the basal forebrain cholinergic function. And my introductory slide here is really the paper that I think indicates where there was a bit of a crossroad in our field. In 2002, we had a science compilation which illustrated this dynamic synapse. And many of the papers described the synaptic failure that was evident in Alzheimer's disease. A couple of years earlier, there was a very interesting paper, High-Level Neuronal Expression of A-beta 142 in wild-type human amyloid precursor transgenic mice by Leonard Mackey and Maslia, and where they described that really the amyloid creates synaptic toxicity without amyloid formation. So in the following years, we were very interested in studying these effects. And whereas many people studied the amyloid aggregation, we started to study this interaction. And in a paper in PNAS from 1999, we studied the cognitive changes of modified processing of amyloid precursor protein in cortical and hippocampal system after cholinergic synapse loss and muscarinic receptor activation. And in these models, we actually deleted the cholinergic neurons by very selective antibody treatment towards NGF, and that kills those neurons. And just to give you a few highlights from that paper, we were interested in the relationship between escape latency in Morris water maze compared to the APP levels in the cortex, and found a correlation such as the more amyloid precursor protein you have, the worse you perform in this task. We continued those studies to try to understand these interactions a little bit more closely. And in the cortical hippocampal APP and NGF levels, we showed that they are dynamically altered by cholinergic muscarinic antagonist or M1 agonist in normal mice, so that sustained increased cholinergic neurotransmission would increase the amount of secreted APP in the CSF. And also interestingly, adaptively reduce the level of total APP, as we showed in the previous paper, and NGF in the cortical hippocampal system. And so the more cholinergic transmission adaptively reduced NGF, and so they were homeostatically linked. And we felt that this could be relevant to understand the pathophysiology then that develops in these diseases. Now, a couple of years later, we decided to put this together in a more theoretical framework in this TINS paper from 2002. And we looked at the dynamic interactions at the hippocampal system, and also added some physiology by having LTP trains in vitro in these hippocampal slices. As you can see on this slide here, when you drive this system, NGF will first increase and then gradually decrease. So there's a relationship between the synaptic activation and NGF levels. Now, taking this system in a framework that describes both the normal physiology and the pathophysiology, we outlined in this 2002 review a system where, as you can see, you have the basal forebrain on the left, hippocampus on the right, and may I note we also had glial cells in this system. So if we start on the right-hand system, the hippocampus on this axis really has several components that are key: APP, NGF, NGF receptors, and in this case, the transmission of the cholinergic system. NGF receptors are transported anterograde, NGF retrograde. In Alzheimer's disease and Down syndrome, and I would say also in the models that are realistic, the basal forebrain, as you know, in Alzheimer's disease, earlier work from Pearson, for example, University of Oxford, and which we replicate in animal models, show loss of basal forebrain neurons, but also their shrinkage. And what was very interesting in these systems that we studied was that we replicated those findings. So on the basal forebrain side, APP is up, NGF is down, or not changed. So colon is down, trachea is down, and P75 NGF receptors are not changed or up in that somewhat homeostatic positive feedback system that is the basal forebrain. In the target zone, APP is up, NGF is actually up because there is lack of retrograde transport. Cholinergic system is down, NGF receptors are down or not changed. But also interesting, we noted this, and this is in reference to, I think, the next speaker too. We noticed large vesicles in large vesicles in the cholinergic system, suggesting that the system is disconnecting and not transporting. Our view of the system at that time in 2002 then indicated that under normal physiological condition, muscarinic activation, cholinergic activation drives the alpha secretase component and drives the secreted form of APP, which had been shown already to be able to interact with NGF and maybe drive that synaptic health transmission. We're of course aware of the beta secretase, gamma secretase idea, but at that point, the field wasn't entirely dependent on understanding just the amyloid plaques. So on the right-hand side, you see what happens in Down syndrome and aging, in that the cholinergic system is less healthy, there's less activation of alpha secretase, and the A-beta component then can generate a different situation. And so of course, in this summary, we also then describe the generation of the A-beta plaque, which is then indicated here by the red stacking, where the field went to remove and what we are perhaps reconsidering in this symposium. Now, a few years later, in 2017, there was very interesting work that described acetylcholine also modulates the gamma frequency of the hippocampus, which we know is really relevant to its function and in cognition. And these M1 receptors, in the same way as we described, were able to entrain that frequency. And as you know, interneurons also play a role in that system. Another interesting article very recently also describes the other end of the spectrum, the other side of the equation, ablation of P75NTR on these cholinergic neurons will also either strengthen or reduce the gamma frequency, showing that even AB-induced degeneration and the dynamics that they saw in this in vitro system are affected by high and low conditions of that system. Now, also to set up for the next talk by Randy Nixon on the potential treatment mechanisms here, several teams, including ours, have worked on the Down syndrome mice, which is remarkably similar to an amyloid precursor protein load, as in trisomy 21. What you can see here is our work from 2005, in which progressive decline of the cholinergic system was apparent over 4 to 12 months. And here you see the 12-month data. There's less cholinergic neuron in the Down syndrome mice compared to Lidomir controls. Now, Bill Mobley has spent a significant amount and of his attention to this model as well. And his recent review in 2019 also support the interpretations and concept we developed in the 2002 article that I described previously. Now, in order to set the scene further, but also to make a twist on this idea, beyond, of course, AD, in Lewy body disease dementia, there are very clear evidence of a role of the basal forebrain system. In fact, the most evident work in this field comes from the work of Allum and so on. But you can see on this recent paper, courtesy of Dr. Lucia Batsu, that this really has a clear role and predicts cognitive decline. So in summary, the reappraisal of basal forebrain function, both in LBD and Alzheimer's disease, really tells us that synaptic changes, whether they are due to various numbers of etiology, are firmly established to control both memory formation and retrieval. The basal forebrain cholinergic synaptic system can regulate the amyloid precursor protein levels. So we can show by various means that the cholinergic system can regulate those. You block it, APP goes up, you activate it, APP goes down. And of course, this relates to the Down syndrome model as well, genetically. So therefore, finally, the renewed interest in synaptic function for the cholinergic system therefore incorporates new views of the cellular interactions of APP in the synaptic space, the various functions they have, the breakdown, as you will hear, the transport of these proteins that can be amyloidogenic, but probably have a very strong normal physiological function and may, when correctly regulated, prevent neurodegeneration. Thank you.
I'm going to be extending some of the concepts that Dr. Isaacson presented and focusing on the particular important role of the RAB5 endosome and its connection to cholinergic neurodegeneration. And these are some take-home messages that are also the outline of what I'll be presenting. In Alzheimer's and Down syndrome, hyperactivation of RAB5, which is a master signaling molecule on endosomes, triggers a direct pathway to cholinergic neurodegeneration. This hyperactivation is driven by AD causative and risk genes, especially APP and ApoE4, and attenuating this hyperactivation blocks cholinergic neurodegeneration. So this is a depiction of the endosomal, lysosomal, and autophagy network. I'll be focusing on the Rab5 endosome. And as most of you probably know, the endosomal lysosomal system is important for internalizing cargoes from the outside or from the membrane and sorting them to different destinations in the cell or degrading them in the lysosome. What's less familiar to many people is the important signaling functions of endosomes, even to the point that the endosome concept includes the concept of a signaling endosome, which has as a platform on the surface many signaling molecules. And for the purposes of this talk, the signaling through neurotrophins to the nucleus is a very key function of this signaling endosome. And of course, there are other roles that are very important for Alzheimer's disease as well. In the late 1990s, Ann Cataldo and my lab was looking at lysosomes and came upon the observation that Rab5 labeled endosomes very well, and that these endosomes were grossly enlarged in Alzheimer's disease. And this preceded amyloid in the cortex when Alzheimer's was identified by the presence of plaques in the hippocampus. Even earlier in Down syndrome, perinatally and then progressively beyond that, this enlargement is evident. It's evident in Down's fibroblasts and in the basal forebrain cholinergic neurons, as well as iPSCs. And as far as we know at this point, it has only been seen in Alzheimer's, Down's, and curiously in Niemann-Pick C1. It's Rab5 mediated, and the enlargement, importantly, slows the retrograde transport of endosomes, and it's signaling by NGF. And it is, as Dr. Isaacson pointed out, is APP dependent. In this case, it is driven by beta CTF, but not A beta. There are a myriad of genes that overactivate Rab5. I mentioned the beta CTF being the most influential, but now there's an accumulation of evidence that that GWAS genes cluster around the function of the endosome, and that many of these, the ones that are in green, actually are either known to activate Rab5 or to increase beta CTF, which in turn we know activates Rab5. And ApoE is one of the most well-established. In addition, proteins that have been known to decrease in Alzheimer's disease involved in endosome recycling, like Rab11 and VPS35, actually would promote enlargement as well, because it reduces cargo exit from the lysosome. So this is some evidence that beta CTF is the essential sole moiety of APP that overactivates Rab5, and it can do it in normal neurons or normal fibroblasts by any number of ways involving a modulation of beta CTF to elevate its levels. And I just point out that gamma secretase, which reduces A beta and increases beta CTF, has the same effect as just solely increasing beta CTF. And this catalyzes what I've called an endosomopathy, consisting of the activation of RAB5, increased endocytic uptake, increased endosomal size, and as I mentioned, the impaired transport of these endosomes that contain NGF signaling as well as other signals. And I point out that there's other evidence that not only APP beta CTF is elevated in AD brains, including sporadic, but BACE and SAPP beta are also consistent with this idea of elevated beta CTF. The converse of what I just showed you is that by lowering beta CTF levels, one can reverse completely the endosomopathy in either human, and I should say human Downs fibroblasts or APP transfected mouse neurons. And to go one step further, deleting one base allele ameliorates the RAB5 overactivation, the cholinergic neurodegeneration. It improves LTP and reverses fear conditioning and Y-maze deficits. One question that was very important to us is how close is the beta CTF to the RAB5 activation? And Sunny Kim in my lab was able to identify that the connection is involves one protein, APPL1. It's unrelated to APP. It has this long name, which is shown here. It's a RAB5 effector that has its own signaling properties, but the most important action here is that APPL1 is recruited by beta CTF to the endosome, and it dimerizes and has the known action of stabilizing the activated form of RAB5 on endosome membranes. And we can see in Downs and AD, the increased recruitment of APPL1 to RAB5 positive endosomes, and APPL1 siRNA reverses endosomal defects in Downs fibroblasts. So removing the APPL1 in this diagram completely reverses the hyperactivation. In addition, a neuron-specific APPL1 overexpression induces the same abnormalities of endosomopathy and cholinergic neurodegeneration that we see in the Downs mouse model. Another important question was whether the action of beta CTF was the essential mediator of all of the downstream effects that we're seeing in this RAB5 upregulated system. And to differentiate beta CTF actions from those of hyperactive RAB5, we generated a RAB5 transgenic mouse expressing RAB5 postnatally, specifically in neurons, at a level that was equivalent to that we see in AD and DS. And in this model, beta CTF levels are normal. So we're looking specifically at the role of RAB5 hyperactivation as a catalyst for downstream pathology. And as summarized here, Anna Pansolfini characterized this mouse, and you can see in pink the number of deficits that are induced by simply overactivating RAB5. And this is dendritic spine loss, LTD especially, and LTP deficits, increased phospho-tau, endosome transport abnormalities that lead to cholinergic loss, and of course, cognitive deficits. And on the right, you see the example of the loss of phenotype of chat neurons that accounts for the decreased number, but also this flagrant neurodegeneration of neurites and cell bodies of cholinergic neurons. Lastly, and this is going to feed into the next presentation, we looked at naflimapimod action on RAB5 overactivation, because we had known from previous work that this drug inhibits the activation of RAB5 as at a very low nanomolar concentration. And when applied to a a Downs model, in this case, TS2 model, very similar to the one that Dr. Isaacson mentioned, it blocks hyperactivation of RAB5 and endosomal enlargement. And most importantly, it rescues the neurodegeneration almost completely back to the wild type level. And as a summary, this slide just reviews the number of manipulations genetically that can hyperactivate RAB5 and lead to this endosomopathy as well as cholinergic neurodegeneration by blocking the transcription of NGF signals via the nucleus. This slide emphasizes that there are now in the literature, including the results that we've presented, that attacking this pathway shown here in any of these various sites is capable of having the same effect of rescuing endosomopathy and rescuing bfcn neurodegeneration in this model, as well as improving cognition. So finally, I would conclude by just reviewing the fact that the genetics of AD strongly support the role of these genes in driving endosome dysfunction. RAB5 hyperactivation drives a direct pathway to cholinergic neurodegeneration via beta CTF of APP, among other genes that can do it more directly. Targeting multiple steps in the RAB5 pathway rescues cholinergic function in vivo, and NFMD, as you will hear, we're providing preclinical proof of concept that targeting RAB5 activation is therapeutic in a mouse model, and you'll hear more clinical evidence supporting this proof of concept. So I'll stop here, and I think this is a good segue to Dr. Allum's talk. Thanks very much for your attention.
This is John Allum. I'm CEO and founder of EIP Pharma, Inc. My title of my presentation, third presentation of the symposium, is Clinical Results with Novel Approaches that Reverse bfcn Dysfunction. This is my disclosure. The therapeutic opportunity in targeting the basal forebrain comes from the knowledge from animal models that the neurodegeneration in the basal forebrain in diseased animals do not actually reflect neuronal loss. Rather, it's a loss of phenotype, or an S and D differentiation of the cholinergic neuron, and that which then no longer function as cholinergic neurons. As a result, in disease models where there's an apparent significant loss of neurons, treatment with, for example, infusing NGF directly, increases and restores the number of cholinergic neurons and essentially reverses disease progression. And there are reasons to believe that this phenotypic loss occurs in humans as well. And then, as Professor Nixon talked in the prior talk, out of the studies on understanding the mechanisms underlying cholinergic degeneration, the two targets that come out for small molecule therapeutics are BACE inhibition and p38 alpha kinase inhibition, and which are the approaches that I'll discuss in my presentation today. With regard to BACE, just one slide. As you in the audience, I'm sure, are aware of the overall results, which show there is not a beneficial overall cognitive effect. Nevertheless, from this paper published this last December, there are reasons to believe that BACE inhibition in terms of the cholinergic neuron does in fact do what it is supposed to do. Specifically, in 2 large phase 3 trials in prodromal AD with distinct BACE inhibitors, there are beneficial effects on executive function, which is generally considered to reflect cholinergic function. What the results imply is that BACE does have the intended effect on the basal forebrain, but perhaps this is offset by globally by negative effects on other aspects of the brain, in particular the hippocampus. Turning to p38 alpha kinase inhibition, I will review the results with our p38 alpha kinase inhibitor in dementia with Lewy bodies, a disease where the basal forebrain degeneration is considered to be the primary pathogenic driver of the dementia. The investigational drug neflamapimod was evaluated in a 91-patient, 16-week double-blind placebo-controlled study. We reported the initial results of this study a year ago at this CTAD meeting, and we'll be reporting further data in this year's meeting. Starting with the effect on cognition, the evaluation of which was a primary objective of the study. On the left, treatment effects on a 6-test neuropsychological test battery designed to assess effects on attention and executive function. There is a significant effect at the higher dose level in the study of 40 milligrams 3 times a day on the NTB that you don't see in the lower dose of 40 milligrams BID. And then on the right, a more specific measure of cholinergic function, an attention composite that assesses effects of drug treatment on the tests within the NTB that evaluate information processing speed. On the attention composite, there's a more robust effect with greater differential and a lower p-value, again in that high dose 40 milligram TID dose group. There is also a significant effect on functional mobility, assessed by the timed up and go test, where there's a significant and clinically meaningful effect if you compare all neflamapimod, including both the low and high dose of neflamapimod versus placebo, and a more significant effect if you look at the 40 milligram TID dose group by itself against placebo. The TUG became relevant to today's discussion because of a series of papers that came out earlier this year, one of which I show on the left, that the gait and balance dysfunction in PD and DLB are due to the basal forebrain disease, that is disease in the cholinergic system rather than the dopaminergic system. There was also a publication earlier this year that demonstrated the direct functional connection between the basal forebrain and a supplemental motor area, the publication on the right. Combined, the literature now indicates that the impairment of gait and motor control and dysfunction we see in DLB, like in cognition, in fact, is likely due to the cholinergic system, which then connects the results that we saw with the TUG test to an underlying drug effect on the basal forebrain. And then perhaps integrating the cognitive and motor effects, new data that we're presenting at this year's meeting from that clinical phase 2 study show that there is a significant effect of neflamapimod relative to placebo on the CDR sum of boxes with a p-value of 0.023, and looking at the mean change at week 16 relative to placebo, there's a 65% reduction with neflamapimod treatment in terms of worsening on the CDR sum of boxes. We're also presenting new data at this meeting on work that we have done to understand the specificity with regard to the drug effect on the basal forebrain. essentially testing this hypothesis that if in fact the drug effect is via actions on the basal forebrain, then the effect should be more specific in patients who have DLB in isolation rather than mixed DLB and AD, where there are other pathogenic mechanisms outside of the basal forebrain cholinergic degeneration in play. To pursue that hypothesis, we leveraged the findings in this paper that came out earlier this year, which shows that plasma phospho-tau correlates with, in particular, tau pathology by PET scan, suggesting that plasma phospho-tau can be utilized to effectively identify patients with mixed AD and DLB and separate those out from patients with DLB in isolation. So working with the laboratory of Charlotte Teunissen at the VU Medical Center in Amsterdam and working with their labs cutoff for plasma phospho-tau 181 for AD copathology, baseline samples from the study were tested, and then the results analyzed by patients with plasma phospho-tau 181 levels less than 2.2 picogram per mil, which would be predicted to be patients with DLB in isolation, and compared those results to those with plasma phospho-tau 181 greater than or equal to 2.2 picogram per mil, which are patients that would be predicted to have mixed AD and DLB. Across the endpoints, the patients with DLB in isolation, shown in purple bars, the effects are substantially better. There's improvement, greater improvement in those patients compared those, and this is compared to placebo, compared to that seen in patients with mixed AD and DLB. And then specifically looking at patients with DLB in isolation, the effects are significant despite small numbers, and the effect size across the board are moderate to large treatment effects versus placebo. The specific effect in patients who should have disease where their disease is limited to the basal forebrain further makes a case that the drug has a very specific and potent effect on the basal forebrain cholinergic system. So what about AD? We had reported data a couple of years ago at this meeting and now have published on the effects of neflamapimod at a dose of 40 milligram BID in Alzheimer's disease, where there was significant reduction relative to placebo on CSF levels of total tau and phospho-tau. On the clinical endpoints at that 40 milligram BID dose level, we did not see an overall effect, but there was an argument within the data and the analyses that had we included a higher dose, as we did in the DLB study, we would have seen an effect, particularly on the Wexler memory scale, which is more of a measure of the cholinergic system, over what we saw in terms of the Hopkins verbal learning test, which is a measure of hippocampal function. So to summarize, rationally designed therapeutic approaches, both BACE and P38 alpha kinase that target specific pathogenic mechanisms of cholinergic degeneration, improve clinical outcomes associated with the basal forebrain, which do provide data that's supportive of the current pathogenic model of cholinergic degeneration that both Ole and Ralph talked about. Therapeutic approaches targeting the basal forebrain hold particular promise in dementia with Lewy bodies, which is consistent with cholinergic deficits being more prominent and widespread in DLB compared to AD. Regarding AD, there's more work to be done fundamentally, but as we can perhaps talk about in the discussion part, there are good reasons to believe that to be optimistic about effects in particular subsets of patients and perhaps in combination with other approaches that target other regions of the brain. Thank you very much, and I look forward to the further discussion.
Gentlemen, thank you for this very, very interesting session. I learned a lot myself, and I was taking copious notes through your presentations. Professor Isaacson, my first question is to you. Which species, is it the oligomeric species that is selectively affecting the bfcn system?
The oligomeric species of what substance?
Of the amyloid.
I don't think so. I think that the basically recycling, as we heard also in Dr. Nixon's presentation, is that the cholinergic synapse depends on a normal functioning transmission. And as I showed in my presentation, our earlier observation of the enlarged vesicles, we didn't know what they were at the time, are consistent with a disconnection syndrome. So when we drove neurotransmission higher through the M1 system, the APP system worked better, if you will. And also as an indicative, NGF was actually functionally reduced, which we always thought reflected the synapse was dynamically adapting. It's a bit confusing because NGF high was supposed to be good, but also indicated that the receptor function that Bill Mobley also showed was decreased. So I think with Professor Nixon's work, it really ties together. And I think the focus is on the dynamic regulation of the synapse. And when it doesn't work, these are the effects that occur downstream.
Very interesting. Professor Nixon, given the fact that the RAB5 is driving early evidence of neurodegeneration, should neflamapimod be applied preclinically and before the onset of symptoms to prevent the degeneration?
Certainly, that is always, I think, theoretically the best strategy. I think what we've shown is that even applying it midstream is able to cut short any further disease progression, at least with respect to things like endosomopathy and cholinergic further degeneration, as well as the tauopathy, which I didn't have time to mention. So the earlier obviously would be better.
Dr. Allum, there's a couple of thoughts I wanted to run by you. First is, would you ever envision a combination of neflamapimod and BACE inhibitors as a combo? And second, would you consider this, once you had induction therapy with a monoclonal, use the combination treatments to prevent further degeneration, and so kind of in a cancer type paradigm?
So I think BACE and P38 alpha kinase inhibition are probably not complementary in that part of the mechanism of P38 alpha kinase, and there's a literature out there both that we've seen in our collaborative work with Ralph Nixon's laboratory, but also in the published literature, that one of the effects of P38 alpha kinase inhibition is actually decreasing inducible BACE expression. So I think there's it's more probably overlapping than a complement, but I think I completely agree that the anti-amyloid beta approaches or other approaches that are targeting synaptic plasticity and hippocampal function, I think are very much could be a complement to our approach of targeting the basal forebrain cholinergic system. I think there's more and more data that we should really be thinking about individual patients and the relative contribution of the basal forebrain versus hippocampal and other pathogenic mechanisms, along with amyloid beta, such as TDP-43, et cetera, which should definitely be combined, in particular in patients with mixed pathologies.
Any other questions that we should discuss amongst us?